So the next little bit is going to be focused on the actual movement of certain substances, either from the tubular fluid or to the blood. So we have a super zoomed in version right here of this portion of the nephron. So here's your glomerulus, part of your capsular space, and this is the opening of your proximal convoluted tubule. So the tubular fluid is going to move throughout, and I'm going to kind of define these in just a second on the next slide, but you can see that things can move across from the tubular fluid into the blood. That is called tubular reabsorption. Or you can see that secretion can happen where you have movement of substances from the blood back into the tubular fluid. So a couple of different ways that can happen. Paracellular transport is where the substance moves in between the cells, just kind of slips in between the cells that are lining the tube. So this is called the tubule epithelium. Another kind is called transcellular transport, where it actually moves through the cell itself, not in between two cells. In order to go through the cell, it has to cross two membranes. It's got to cross this one, and it's got to cross this one, and then it can get into the blood or vice versa. Okay, so that's basically what I just said. In order for a substance to move from the tubular fluid back into the blood, or it could go the other way around as well, it could go between the cells, which is paracellular transport. It can go through the cells, which is transcellular transport, in which case it's got to go through the two membranes, the luminal and the basolateral membrane. It can cross by using transmembrane proteins. So transmembrane proteins, I think of like little gates. The gate has to be there and it has to open in order for the molecule to cross over. It can't just diffuse across the membrane. Or it can go by bulk flow. So bulk flow, remember, is where we get kind of a relatively large amount of fluid and whatever solutes are dissolved in it, and they all move together. And that can happen here because the peritubular capillaries have a low hydrostatic pressure and a high osmotic pressure. So make sure you understand why that's the case. If you kind of backtrack from where the peritubular capillaries came from, so let's go all the way back to that afferent arteriole that feeds into the glomerulus. So you go into the afferent arteriole, which leads into the glomerular capillaries. Well, what process is happening there? That's glomerular filtration. So we're losing fluid and solutes out of the blood vessels in the glomerular capillaries, and then it leaves through it leaves through the efferent vessel and then continues on to the peritubular capillaries. So while we were still in the glomerulus, we lost a lot of fluid and small solutes, but there was a lot of things that didn't get filtered out because we certainly don't want to. So we've got a lot of large proteins in there. So by the time you make it to the peritubular capillaries, you have a low volume and a high concentration of proteins. So that low volume gives you the low hydrostatic pressure, and the high concentration of proteins gives you the high osmotic pressure. So the combination of those two things make those peritubular capillaries want to draw fluid and solutes back in by bulk, and that's going to be kind of bulk flow movement. So let's talk about the transport maximum and the renal threshold. So these are really two important concepts in terms of movement of solutes out of the tubule fluid and back into the blood. So transport maximum, abbreviated TM, is the maximum amount of a substance that can be reabsorbed or secreted, so we can go either way, across a tubule epithelium in a given period of time. So the maximum is going to be dependent on the number of transport proteins in an epithelial cell membrane for a specific substance. So transport proteins are what I just referred to as like little gates. If only one person can go through a gate at a time, then the number of gates limits how many people can move from area A to area B. So that's kind of what we're looking at. So let's look at it from a specific example. So we're going to use glucose. So the transport maximum for glucose reabsorption, so we're talking about getting glucose from the tubule into the blood. So let me kind of backtrack for a second. We're going to filter nearly all of our glucose. So as the blood is going through the glomerular capillaries, we're going to filter nearly all of that. Having said that, we don't actually want to excrete all of that. We're going to end up bringing it back in through this tubule reabsorption. So it's critical that we get it all back in. Okay. So the transport maximum for glucose is approximately 320 milligrams per minute. So as long as your tubular fluid is within kind of normal homeostatic range, and it doesn't have more than 320 milligrams of glucose passing through that region any given minute, then all of that glucose is going to be absorbed. If our tubular fluid glucose levels are high, and in any given minute we have more than 320 milligrams passing through it, anything above 320 milligrams doesn't get reabsorbed. It's going to continue on through the tubule system, and it's going to end up in the urine. So it's going to end up getting excreted in the urine. So if we have a normal blood sugar, then we should have 320 or less milligrams of glucose passing through our tubular fluid in any given minute, and it should get all completely reabsorbed just fine. If our blood sugar is elevated and then we filter all of that out in the glomerulus, then we end up with an elevated glucose concentration in the tubule fluid, and then we have so much passing through at any given moment or at any given minute that we can't absorb it all, and then we end up excreting it. So that's why you get glucose in the urine. So a related concept is renal threshold, and I kind of just touched on that. So transport maximum refers to actually in the tubule system. So we're talking about how many molecules of glucose can actually get absorbed in the tubule system per any given minute. Renal threshold kind of takes that and relates it back to the plasma concentration. So the renal threshold is the plasma concentration of a substance that can be carried in the blood without eventually appearing in the urine. So it's basically how much level of this substance can we have in the blood without exceeding our transport maximum. So sticking with our glucose concentration, 220 milligrams per deciliter is going to be our renal threshold. If your blood sugar is above this amount, then by the time it gets filtered in the glomerulus, you are going to exceed your transport maximum. You're going to get past that 320 milligrams per minute, and then it's going to show up in the urine. Okay, so transport maximum, we're looking at what's going on in the tubule system. It's basically a rate, so milligrams per minute. Renal threshold, we're looking at a plasma concentration. So we're going to be looking at milligrams per deciliter. So what our renal threshold is, what is our plasma concentration of any given substance that we can have without exceeding our transport maximum that's going to take place in the tubule system. So we talked about what happens if glucose shows up in the urine. So that's called glucosuria. Glucosuria is the excretion of glucose in the urine. This is going to occur when the plasma glucose levels exceed 220 milligrams per deciliter. So we've exceeded our renal threshold, and the transport maximum for glucose has been exceeded, because those two kind of go hand in hand. So glucose molecules act as an osmotic diuretic, pulling water into the tubular fluid and causing an excessive loss of fluid in the urine. Glucosuria, along with frequent urination and thirst, are classic signs of diabetes mellitus. So you may have heard that, people with untreated diabetes, they're really, really thirsty a lot and they urinate a lot. And kind of like your first thought would be they're super thirsty, so they're drinking a lot, and that's why they're peeing a lot. In reality, what's happening is almost kind of the reverse situation. So they have excess glucose in their blood, right? And then that translates to having excess glucose in their tubular fluid. So they're maximizing they're exceeding their transport maximum. So they have glucose remaining in the tubular fluid. So that extra glucose in the tubule fluid acts as kind of this osmotic agent, and it pulls more fluid into the tubule system. So it draws more fluid in, so it increases the amount of urine they produce. So they're actually excreting excess urine, and that's in turn what's causing them to be thirsty. So it's not that it's kind of a chicken and the egg. It's not that they're thirsty first and so they urinate more later. It's the extra glucose in the tubular fluid that makes them urinate more, and that in turn leads them to feel thirsty. Some substances are not normal components of urine because we reabsorb 100% of them. So glucose was one of those that I talked about. We're going to filter it freely, but as it goes through the tubule system, we're going to make sure that we recapture all of that glucose through reabsorption. So normally this is going to happen in the proximal convoluted tubule. If it's going to be 100% reabsorbed, it happens in the proximal convoluted tubule. There's two main kind of substances that we would kind of fit into this group. One of those are nutrients, so not only glucose, but amino acids, things like lactate, so nutrient molecules, because it really doesn't make sense to get rid of nutrient molecules in the urine, because we can always use nutrient molecules, to build things up in our body. And then the small amount of filtered plasma proteins. So remember I said the majority of proteins don't get filtered. Usually they're too large, but if we do have small plasma proteins, they do get filtered, but they usually get completely reabsorbed back in the proximal convoluted tubule. So let's look at, we're sticking with glucose. Glucose is kind of our classic example. Nutrient reabsorption, so this is going to be the ones that are completely reabsorbed in the proximal convoluted tubule. You can see that up here. Okay, so glucose is first transported into the tubule cell across the luminal membrane by the sodium glucose symporter. So let's look at this diagram. So here we are in the proximal convoluted tubule. We're going to zoom in. This is our tubular fluid. This is our tubule epithelial cell. This is the interstitial fluid, which is a small space between the tubule cell and the blood vessel, and then this is your peritubular capillary. So when we talk about tubular reabsorption, we're talking about something going from the tubular fluid back into the blood. Okay, so for glucose, the first thing that happens is it's going to be taken up by the sodium glucose symporter. So symporter means two molecules are going through that membrane protein and they're going in the same direction. So sodium is going to be high here in the tubular fluid, and glucose is actually going to be higher in the cell. So you have sodium and glucose kind of going through the transport protein, and then they end up in the tubule cell. So the energy from sodium moving down its concentration gradient, so sodium's going from high to low. So the kinetic energy of sodium moving down its concentration gradient is going to be harvested to push glucose against its concentration gradient. Glucose is going from low to high. It doesn't want to do that. Everything usually likes to go from high to low, but if we have something else going from low to high, I'm sorry, I just said that wrong. If we have another substance going from high to low, like sodium, we can use that energy to draw the other one with it from low to high. That's called secondary active transport whenever we do that. So the sodium and the glucose go together, and then you end up in the tubule cell. Now glucose will diffuse across, and then you have glucose uniporters on the other membrane that will simply move it out of the cell. And then from there, it's just going to diffuse across and get absorbed into the bloodstream. Okay, so glucose is ultimately going to get returned to those peritubular capillaries. Peritubular, because it's the capillary that's right next to the tube. And then just like any other substances that rely on membrane transport proteins, there's going to be a maximum amount that can be absorbed per unit time. So we've got sodium glucose symporter, we've got the glucose uniporter. We cannot use these beyond their capacity. We only have a certain limited number of these protein molecules, so that's going to be kind of our rate limiting step. Some substances are recovered completely from the tubular fluid, but others are not going to be completely reabsorbed. So sometimes they will, sometimes they won't, depending on certain factors. So if it's not completely reabsorbed, that means that some of it's going to end up being excreted in the urine. So by varying the amount of a certain substance that it's excreted, the nephron then has a substantial role in regulating the blood level of it. So it's kind of up to the nephron. How much, well, it's kind of up to the nephron and the tubule system. How much of this substance are we going to let stay in the body, and how much are we going to excrete with the urine? So some of the things that fall in that category: sodium, water, potassium, bicarbonate, and calcium. So these all look like pretty important things. We've seen along the way some important roles for these molecules. So this is going to, by and large, depend on what's already in our body. If we already have enough water, if we're well hydrated, we don't need to keep reabsorbing it and retaining more and more. So we are free to allow any extra water to get excreted in the urine. And same with all the other molecules. So it's really going to depend on the state that our body is in relative to whatever these molecules are. So sodium is kind of unique because sodium gets reabsorbed all along the nephron tubule. A lot of things are either specific to, it gets mostly reabsorbed in the proximal convoluted tubule, or just the nephron loop, or just the distal. But sodium kind of gets absorbed all along the way, although the way it does so changes depending on where in the tubule system you are. So again, the sodium concentration is high in the tubular fluid and in the interstitial space, but it's low in that tubular cell. So remember, if you picture that tubule cell, on the top of it was the interstitial space, on the bottom of it was the tubule inside the tubule system. And so in both of those spaces, the sodium concentration is high. In the actual tubule cell, it's low. So why is that? Once again, we come back to the sodium-potassium pump. Sodium's always pumping the cell is always pumping sodium, out of the cell. So we're constantly pumping that sodium out, which keeps our cellular sodium level low, but it makes it high in the tubular fluid and in the interstitial space. So sodium can passively cross the luminal membrane into the tubule cell. That's kind of like what we saw with the glucose. We saw it was moving from high to low. It just wanted to go into the cell. So it makes its way into the tubule cell, but then the sodium-potassium pump actually pumps it out the other side of the cell. So at that point, then it enters the peritubular capillaries or the vasa recta, depending on where in the tubule system we are. So again, from the tubular fluid, it goes through like a sodium transport protein nice and easily because it's moving down its concentration gradient from high to low, gets into the cell. Once it's in the cell, it can diffuse across to the other side of the cell where the sodium-potassium pump will pump it out. And at that point, it's pumping it into the interstitial space, and it'll get reabsorbed by either a peritubular capillary or a vasa recta capillary. Okay, so considering that sodium intake can vary between individuals, it can be as little as 0.05 grams per day if somebody's on a low sodium diet, or 20 to 25 grams per day if somebody's not on a low sodium diet. So because the intake varies, our output is going to be critical to maintain our sodium levels in the blood where they need to be. So aldosterone is a steroid hormone produced by the adrenal cortex. We have briefly talked about this before. It stimulates an increase in the synthesis of sodium channels and sodium-potassium pumps in the tubule cells. And that allows for more reabsorption. Kind of aside here, water always follows sodium. So anywhere sodium goes, water is going to go. And then potassium gets secreted as a result. So let me stop and walk y'all through that again. So aldosterone is a hormone that gets produced by the adrenal cortex. One of its job is to go to the tubule cells and increase the amount of sodium channels and sodium-potassium pumps in the tubule cells. So anytime you increase those proteins in the tubule cells, you're going to allow more sodium to get reabsorbed out of the tubular fluid and into the blood. Okay, so ultimately aldosterone increases the amount of sodium reabsorption. So how does that affect water and potassium? Water loves sodium. If sodium's going back in the blood, water's going to follow it. It's like its little buddy that likes to tag along. Potassium gets secreted. Okay, so this is a little tricky. If sodium is getting reabsorbed, it's going from the tubular fluid to the blood. And this tells you that potassium is getting secreted, meaning potassium is going from the blood to the tubular fluid. Why is that? That's because of the sodium-potassium pump. Anytime the cell uses that pump to push sodium from the inside of the cell out towards the blood, it has to take potassium out of the blood and push it back into the cell because they always move in those opposite directions. Once the potassium gets into the cell, it's going to kind of diffuse out the other side and it's going to end up in the tubular fluid. So because of the sodium-potassium pump, because it's always moving those two molecules in opposite directions, anytime sodium gets reabsorbed, potassium is going to end up getting secreted. And once it gets secreted, it's in the tubular fluid, and it's going to get excreted with the urine. Okay, atrial natriuretic peptide, or ANP, this one inhibits sodium reabsorption by increasing glomerular filtration rate. So this is what I talked about in part 1 of the video. Anytime we increase the glomerular filtration rate, it speeds up the rate at which the fluid travels through the tubule system. And if it's moving through the tubule system very quickly, we don't have enough time to reabsorb everything. So it's just by nature going to increase the amount of fluid and solutes that appear in the urine. So aldosterone increases sodium reabsorption. And then I kind of always keep in mind when you tell yourself that water is going to follow and potassium is going to go the other way. And atrial natriuretic peptide, or ANP, is going to inhibit sodium reabsorption, and it's going to do that by increasing the glomerular filtration rate. So this kind of just shows you that movement of sodium. It's broken down into, this first panel is showing you what's happening in the proximal convoluted tubule, and the second panel is showing you what's happening in the distal convoluted tubule, collecting duct, and or collecting tubule. So one of the important distinctions to make is that reabsorption that's happening on the early end in the proximal convoluted tubule is sort of unregulated. It's just kind of doing what it pleases. Everything that's happening at the distal convoluted tubule, collecting duct, or collecting tubule, this is mostly regulated by hormones. Okay, so looking in the proximal convoluted tubule, you can see the sodium. It's coming in through its transport protein. It then diffuses across the other side of the cell into, here's your sodium-potassium pump, and it pumps sodium out of the cell. Consequently, the potassium moves into the cell. Over here in the distal convoluted tubule, here's your tubular fluid, here's your tubule cell, here's your interstitial space, and then here is your peritubular capillary. So the main purpose of this image is just to show you that this is regulated by hormones. You've got aldosterone here. Aldosterone increases the amount of sodium channels, shown here, and sodium-potassium pumps, shown here. So whenever aldosterone gets released from the adrenal cortex, it's going to come down here to the distal convoluted tubule. It's going to put a bunch of these proteins on the tubule cells, and that allows more sodium to move out of the tubular fluid and into the peritubular capillaries. So water reabsorption happens through osmosis and generally is following the movement of other solutes and ions. It can go across the cellular layer by paracellular transport, where it's kind of slipping in between the cells, or by transcellular transport, where it's actually going through the cell. If it's going through the cell, a lot of times that happens through aquaporins. Aquaporins are little water-filled channels that get inserted on the plasma membrane, and actually most of the water that gets reabsorbed goes through these aquaporins. So it's kind of like, we see these transport proteins for other molecules, and aquaporin is kind of like a transport protein for water. It's just a water-filled channel that gets inserted on the plasma membrane of the tubule cell and allows water to move through the cell more easily. So the majority of the water reabsorption happens in the proximal convoluted tubule as a result of the water following sodium. We saw that a lot of the sodium was reabsorbed there, and I said that water likes sodium, so wherever it goes, it's following. So a lot of water reabsorption happens in the proximal convoluted tubule because it is simply tagging along with sodium. That is called obligatory water reabsorption. It's just going to happen because sodium's moving in the direction that it wants to move, and so water's just going to tag along with it. There's a small amount of water that gets reabsorbed in the nephron loop. Probably the least amount of water gets reabsorbed in the nephron loop. And then you get other water reabsorption that happens in the distal convoluted tubule. And like I mentioned in the previous slide, that is going to be regulated by hormones. That's called facultative water reabsorption because it's happening as the result of some sort of hormonal changes. Okay, so let's look at some of the hormones that would affect this. Aldosterone. Okay, so we've talked about aldosterone. What did it do? It increased the amount of sodium channels on the tubule cells in the distal convoluted tubule, which means that sodium reabsorption increased. So if sodium reabsorption increased, water reabsorption is going to increase because water is going to follow sodium. So even though aldosterone really directly only affects sodium, it kind of consequently affects water because water is going to move where sodium moves. The other one is antidiuretic hormone, or ADH. We've seen this. It comes from the posterior pituitary. ADH has a more direct effect on water because it causes an increase in the expressions of aquaporins on the principal cells that are there in that disconvoluted tubule. And so that's actually adding more of the aquaporins, which is more of a direct effect. It allows more water to move across in the distal convoluted tubule. So you get more water reabsorption. So obligatory water reabsorption is happening in the proximal convoluted tubule simply as a result of water following sodium. And again, sodium's just kind of moving where it wants to. It wants to leave the tubular fluid and go back into the blood just based on concentration differences. A little bit happening in the nephron loop. And then you've got more happening in the distal convoluted tubule. What's happening there is called facultative water reabsorption, and it's called that because it's dependent on hormonal state. So changes in hormone levels, specifically aldosterone and ADH, is what regulates the increase or decrease, well, in this case, the increase of movement of water from the tubular fluid back to the blood. Aldosterone directly impacts sodium. It adds more sodium channels, so more sodium gets reabsorbed, consequently more water gets reabsorbed. If it's ADH, it adds more aquaporins, so it directly allows more water to move out of the tubular fluid into the blood, so more water reabsorption that way. So this is just showing you the effects of the antidiuretic hormone. So these little guys are your aquaporins, and anytime you add ADH, you get a release of ADH from the posterior pituitary. So you can see these are like aquaporins that are sitting in a vesicle. And this happens with a lot of our membrane proteins. We make them in bulk amount, but we don't need them always in bulk amount. So we kind of put them in a vesicle and we leave them here. So they are like ready to be dispersed when needed. But they're not just always going to be here, because if they're always here, we're going to have constant high water reabsorption. So anyways, here are our aquaporins, and they are ready and waiting in these vesicles. And when ADH comes out, it stimulates these getting actually put into the membrane. So we get more aquaporins along the membranes of our tubule cells, and that allows water to easily flow out of the tubular fluid and then back into the blood. Okay, so antidiuretic hormone, it increases the water reabsorption from the filtrate into the... Okay, so antidiuretic hormone, increased water reabsorption. So let me cross this word out, filtrate. This should actually be tubular fluid, because we know that filtrate is only while it's in the glomerulus, right? So ADH increases water reabsorption from the tubular fluid into the blood. And what that's going to do is it's going to create a much smaller volume of urine, and that urine is going to be more concentrated because all the water went from what would be the urine back into the blood. Conditions of extreme dehydration result in high levels of ADH. So if you're not taking much fluid in at all, you certainly don't want to be excreting any in the urine. So you're going to have very high levels of ADH so that you can move as much of the water from the tubular fluid as possible back into the blood. So the urine can be as little as half a liter per day. Under those circumstances, the urine is going to be very dark and concentrated. A decrease in ADH results in an increase in urine volume. So if you don't have ADH around, moving more of the water out of the tubular fluid into the blood, it's going to stay in the tubular fluid. It's going to create a larger volume of urine, and it's going to be lighter in color and less concentrated. So you can have several liters of urine being produced and released each day if you don't have a lot of ADH around, which would be the case if you're, well hydrated. So potassium movement is kind of unique. It varies depending on where you're at. And so I kind of gave you an example already about one way that it can move, and it was in opposition of sodium because of the sodium-potassium pump. But that's not always the case. So in the proximal convoluted tubule, about 60 to 80% of the potassium is in the tubular fluid, and that's going to get reabsorbed and back into the blood by paracellular transport, kind of again dependent on the sodium movement. So these steps 1 through 4 kind of explain how and why potassium is moving from the tubular fluid back into the blood. So sodium gets reabsorbed. We know that. Sodium likes to go from the tubular fluid back to the blood. So sodium gets reabsorbed, water follows. So now we have a large movement of sodium and water out of the tubular fluid into the blood. The consequence of that is that the concentration of the remaining solutes in the tubular fluid increase. Because we're losing so much water, we're concentrating what's staying in the tubular fluid. So now we have a gradient where you've got kind of more water in the interstitial space area than you do in the tubular fluid, and that's going to draw more solutes that direction. So you're drawing more solutes towards the blood, and so potassium can move that way through the paracellular route, going out of the tubular fluid and into the blood. This is also going to be the case for other things like magnesium, calcium, phosphate, fatty acids, and urea. About 10 to 20% of the potassium in the tubular fluid gets reabsorbed along the thick segment of the nephron loop ascending limb. You can either use transcellular or paracellular transport. Either a net reabsorption or a net secretion of potassium could result in the collecting tubule. So we have specialized cells called type A intercalated cells that are going to continuously reabsorb potassium. And then we have other cells called principal cells that are responsible for potassium secretion, but that's going to happen at varying levels based on aldosterone levels. So again, aldosterone increased the amount of sodium channels, so it directly affects sodium, but as aldosterone increases those sodium reabsorption, the consequence is going to be the potassium secretion because of the opposing movement due to the sodium-potassium pump. So that was kind of what I explained earlier. So here in the proximal convoluted tubule, so I would kind of look at this, like kind of read the 2 slides before it, but kind of look at this, it kind of lets you visualize it instead of just bulk information and words. Proximal convoluted tubule, you have the majority of potassium movement. In this case, it's potassium reabsorption. So this is the one where the movement of sodium led to the movement of water. So we got this kind of dilution of the area outside of the tubule, and that drew the potassium back to that dilute area. So it drew it out of the tubule into the blood. 10 to 20% in the thick section of the ascending limb. Okay, so here's our descending limb, thick segment, and then thin, ascending limb, thin, and then thick. So 10 to 20% of potassium reabsorption is going to happen here in the nephron loop. And then here's your distal convoluted tubule, collecting tubules and collecting ducts. You've got regulated calcium reabsorption. Regulated meaning it's going to be based on hormonal changes. Here you have type A intercalated cells that are going to continuously reabsorb potassium. And then you also have principal cells that are going to be responsible for potassium secretion, but how much it does that is going to depend on aldosterone levels. Higher aldosterone levels lead to higher sodium reabsorption, lead to higher potassium secretion. And then just keep in mind, when you talk about secretion, if something's being secreted, that means it is ultimately going to be excreted with the urine. Calcium exists in the blood on its own. It can just be free calcium. But whenever phosphate, so this PO43-, that's phosphate, whenever phosphate levels are high, calcium likes to combine with it and join together, and they form calcium phosphate, which is a mineral, and then that gets deposited into bone. So calcium is fine to be free on its own, but when you have high levels of phosphate, it prefers to bind up with it, and then that combined molecule, calcium phosphate, goes to the bone. But whenever the phosphate levels stay low, calcium will just stay in the blood on its own. So the amount of calcium and phosphate that get excreted in the urine is regulated by parathyroid hormone. So parathyroid hormone, we've talked about this, it comes from the parathyroid gland. And the way it works is parathyroid hormone gets released from the parathyroid gland whenever our blood calcium levels start to decrease. So when our blood calcium level is going down, parathyroid hormone gets released, and one of the places it's going to go is to the kidney tubules. So it will inhibit phosphate reabsorption in the proximal convoluted tubule, but it will stimulate calcium reabsorption in the distal convoluted tubule. Okay, so stimulating calcium reabsorption makes sense because our blood calcium level is dropping. We want to reabsorb more, so we bring more calcium out of the tubular fluid into the blood. That makes sense. Why would we want to inhibit phosphate reabsorption? Well, I just said when phosphate levels are high in the blood, calcium wants to bind up with it. And it creates calcium phosphate and it goes to the bone. So you didn't do anything to increase your blood calcium level if all it's going to do is bind to phosphate. So we're going to inhibit phosphate reabsorption. So phosphate is going to stay in the tubular fluid and get excreted in the urine. And so we've got more calcium coming in, getting reabsorbed from the distal convoluted tubule, but without any phosphate around to bind it up, it's going to stay in the blood and it's going to bring our calcium levels in the blood back up. So here you can see that parathyroid is inhibiting the reabsorption of phosphate in the proximal convoluted tubule. So this is where phosphate would normally be reabsorbed, but if parathyroid hormone is there, we're going to inhibit that. So phosphate is going to stay in the tubular fluid and ultimately get excreted with the urine, but parathyroid hormone is going to stimulate the reabsorption of calcium in the distal convoluted tubule. So we're getting more calcium in the blood and less calcium in the urine, but we're getting less phosphate in the blood and more phosphate in the urine. And so that allows the calcium to move into the blood but stay free as opposed to binding to phosphate. And so our free calcium will then work to increase our blood calcium level. Okay, so bicarbonate ions, hydrogen ions, and pH. So anytime we're talking about hydrogen ions, we know it's going to affect pH, and in this case we're talking about the pH of the blood and the pH of the urine. Now keep in mind the pH of the blood has to stay in a very narrow pH range, whereas the urine pH has a very broad range of acceptable levels. Okay, so bicarbonate, which is this HCO3-, this is a weak base. It acts as a buffer, as we'll see in future lectures. But it kind of offsets hydrogen. So if you were to add this hydrogen ion to this bicarbonate, it becomes H2CO3, which is something called carbonic acid. So this kind of neutralizes the hydrogen ion, because we know that hydrogen ions are what make solutions acidic, right? Okay, so keep that in mind. And here we're talking about what happens as they go through the filtration membrane. Bicarbonate ions move freely across the filtration membrane. So that means you're going to end up with a bunch of bicarbonate in the tubular fluid. But only a small amount of the hydrogen ions get filtered. So the majority of the hydrogen ions are going to stay in the blood. So when those two came in the afferent arteriole into the glomerulus, they were together. So everything was nice and neutral. But as it went through the glomerulus, the bicarbonate got filtered, but the hydrogen did not. So now they're separate. So what do you think that suggests about the blood that's leaving through the efferent arteriole? It's going to be kind of acidic because it has a lot of the hydrogen ions. So what that means is we have to very quickly get that bicarbonate back into the peritubular capillaries, thereby the proximal convoluted tubule, so that the blood doesn't really become and stay too acidic. So about 80 to 90% of the bicarbonate gets reclaimed, and I use that term because it's not a direct reabsorption, as you'll see in the next slide. But the majority of it gets reclaimed from the tubular fluid, primarily in the proximal convoluted tubule. The remaining 10 to 20% gets taken up by the thick segment of the ascending limb of the nephron loop. So as the tubular fluid enters the distal convoluted tubule, by that point, normally all of the bicarbonate that was originally filtered has been reabsorbed or reclaimed. And again, we need that because the hydrogen didn't get filtered. So it would cause acidity of the blood if we don't hurry up and get that bicarbonate back to it. So this slide shows you how we get the bicarbonate back into the blood. It's a little bit complex. We'll just call it interesting. So here's the tubular fluid, here's your tubule cell, here's your interstitial space, and here's your peritubular capillary. So remember, after filtration, we had a bunch of hydrogen ions in the peritubular capillaries, but a bunch of our bicarbonate was in the tubular fluid. So we need to get this bicarbonate back to the hydrogen so that we don't get too acidic. Okay, so it's kind of step by step. With step number 1, within the filtrate, HCO3-, that's bicarbonate, it combines with hydrogen to form carbonic acid, which is H2CO3. This is unstable, so it dissociates to form carbon dioxide and water. So here's your bicarbonate. It will find a hydrogen molecule. Those join together to form bicarbonate, H2CO3. This dissociates or comes apart, but it doesn't come apart to make what it originally was, bicarbonate and hydrogen. It comes apart to make carbon dioxide and water. Okay, step 2, carbon dioxide goes into the tubule cell by simple diffusion. Step 3, we basically reverse the reaction that just happened. Carbon dioxide is going to combine with water to create carbonic acid, or H2CO3, and then carbonic acid is going to dissociate. We're going to lose a hydrogen ion. The hydrogen ion is going to come back in here, and we can participate in further reactions, and it's going to leave bicarbonate, or HCO3-, which is then free to diffuse across, and it's going to join the other hydrogen back in the peritubular capillaries. So this is what's going on in the proximal convoluted tubule. If you look at what's happening in the collecting tubules, you have, this is your type A intercalated cell. Step 1, you have carbon dioxide diffusing in from the tubular fluid into the tubule cell. Carbon dioxide combines with water to form carbonic acid, or H2CO3. From there, we're going to lose a hydrogen. It's going to come back into the tubular fluid, and that when you lose that one hydrogen, it gives you HCO3, which is your bicarbonate, and then that can simply diffuse across back into the blood of the peritubular capillaries. So we've talked a lot about how things get reabsorbed because our body needed them and didn't want it to get excreted in the waste. But some of the things, a lot of the things we do need to excrete. That's kind of one of the purposes, is so that we can get rid of things that might become toxic if accumulated, or just things that our bodies don't need that we need to get rid of. So the urinary system prevents the accumulation of that cellular waste, various hormones or metabolites, foreign substances like drugs and chemicals that we may take into our body. We can get rid of them with the urine. Materials that are going to be excreted in the urine can either be filtered at the glomerulus and it just stays in the tubular fluid the whole time, or if it didn't get filtered, it can get secreted along the tubule pathway. Either way, as long as it ends up in that tubular fluid, then it's fine to get eliminated with the urine. Some of the things that you might see being eliminated: urea, uric acid, creatinine. Those are all metabolic byproducts from liver and muscle. So the liver and muscle cells are going to create those as they do their normal metabolic reactions, but we don't want those accumulated in the blood. So those are things that are going to get filtered out or put in the tubular fluid so that we can get them excreted with the urine. Certain drugs, so antibiotics, morphine, marijuana, things like that, that get in our system, we've got to get them back out. So those will also be excreted with the urine. And then hormones. Human chorionic gonadotropin, or HCG, is a hormone that gets produced by pregnant women only, and then it gets excreted in the urine. So that's how we can use a urine analysis to check for a pregnancy. If that HCG is excreted in the urine, it would only be doing so if the individual was pregnant. So we can use that for a rapid pregnancy test. So we spent a lot of time talking about glomerular filtration rate, how we regulate it using different mechanisms, and the importance of it in terms of, if it's too high, we end up excreting necessary substances in the urine. If it's too low, maybe we don't get enough excretion and we can get a buildup of toxic substances. So one way that we have to assess kidney function is to actually measure that glomerular filtration rate. So to do this, they inject a person with something called inulin. Inulin is a polysaccharide derived from plants. It gets freely filtered in the glomerulus, but it doesn't get reabsorbed or secreted. So as that inulin travels through the glomerular capillaries, it should be completely filtered. But unlike other substances, once it gets into the tubule system, once it enters that proximal convoluted tubule, whatever was filtered is not going to change. It's not going to get reabsorbed. And if it didn't get filtered, it's not going to get secreted. So we can then go back and measure how much inulin is being excreted in the urine, and that will tell us how much was actually originally filtered. So they give each individual enough inulin for them to achieve a plasma concentration of 1 milligram per milliliter. And then the urine later is going to be collected and measured for volume and the concentration of inulin. And then in addition to that, they'll draw blood and monitor the plasma concentration at different time intervals as well. So from all of that information that's collected, you can get a glomerular filtration rate. The normal rate for an adult individual is going to be 125 milliliters per minute. That's how much we should be filtering our plasma as it goes through those nephrons. A lower glomerular filtration rate is indicative of a reduced kidney function. And so if your kidneys are not filtering at an appropriate rate, you're more likely to get nitrogenous waste products and other unwanted substances that are going to accumulate in the blood, because they should be getting filtered out. But if we got a rate that's too low, then we're not filtering enough out, and then that's just going to accumulate in the blood. So this slide talks about renal failure or kidney failure, some of the treatments in terms of dialysis and a kidney transplant. So I'm not going to read through all the details. I'm going to let you guys kind of read through everything on your own. If I can just kind of give you the gist of it, renal failure is basically when almost 100% of your kidney function is reduced. You're getting like 80 to 90% reduction in kidney function. This could be a result of a number of different things, certain diseases such as diabetes, high blood pressure, autoimmune disease where it's actually attacking some of the kidney cells. There's a variety of things that can contribute to renal failure. So one of the ways that we can kind of treat it, I say treat it, it's not really a treatment because we're not fixing anything, but we can utilize dialysis where you kind of get hooked up to a machine and the machine will sort of do the job of the kidneys for you. It pushes your blood through there and it removes the unwanted waste products that would otherwise get removed during normal glomerular filtration and then kind of puts the blood back into your system. But again, I'll let you kind of read the details on dialysis. The other alternative is a kidney transplant, which obviously is, where you get a healthy kidney from somebody else. One of the key points that I don't think most people know is when they do a kidney transplant, they don't remove the diseased kidneys. I suppose sometimes they would if it were some kind of metastatic cancer and they really posed a threat to the body. Certainly they would take it out, but in most cases the kidneys are just not functioning. They're not necessarily harmful, they're just not working well. So what they'll do is they'll take the new kidney and they'll just add it in. You can see the diseased kidneys are up here, they're not working too great. They take the healthy one and they'll just kind of attach it in lower in the abdominal pelvic cavity and kind of give it a vascular connection and attach a ureter to it, and then you're good to go with your transplanted kidney. Okay, same thing here. I'm going to let you guys kind of read through this on your own. This is talking about renal calculi. That's kidney stones, which is basically a mineralization, mostly of calcium salts that get deposited in the kidney. And the problem is when they start to go through the ureter, it is incredibly painful. The ureter is a very small fibromuscular tube that's really meant only for the passage of urine. So when you get this kind of clumpy crystalline structure that tries to go through there, it's very painful. So they refer to it as loin to groin pain as it starts up in the kidneys and moves all the way down to the bladder. The pain does subside sometimes when it gets in the bladder just because at that point there's more space. Some people still report pain while it's in the bladder, and then it passes through the urethra. So anything 4 millimeters or less can pass on its own. It's still going to be excruciatingly painful, but it can pass on its own if it's 4 millimeters or less. Anything above that, they'll need to go in and break it up. They can do that with something called a lithotripsy, where they go in and they use shock waves to kind of break it down into smaller pieces, which then pass. Alternatively, they can insert a scope through the urethra into the bladder, and however far they need to go, again, to try to break it up to allow smaller pieces to pass. Okay, general characteristics about urine. Urine's a fluid that exits the kidneys. It's going to go through the ureters. It's going to be stored in the bladder. And then when it's time for excretion, it's going to go from the bladder through the urethra where it'll be eliminated. That process of elimination is called urination. It's also called micturition. It's going to be a sterile excretion unless you've got contamination with some kind of infection in the kidney or along the urinary tract. Now, of course, if it were to exit the body and stay somewhere for a while, it wouldn't stay sterile very long, but as long as there's no urinary tract infection, it should be sterile as it exits the body. Typical urine composition is about 95% water and then 5% solutes. You can see some of those various solutes here: sodium, chloride, potassium, magnesium, some of just general salts. Nitrogenous waste, so urea, uric acid, creatinine, these are nitrogenous-based waste. Hormones, we talked about those. Drugs, we mentioned. And then small amount of ketone bodies, and ketone bodies are one of the waste products that gets formed when you digest fatty acids. The average daily volume of urine is going to be between 1 to 2 liters. This is going to vary depending on a variety of different factors. So how much fluid you're taking in. Obviously, the more fluid you take in, the more fluid you're going to put out. Blood pressure. We know that pressure and volume are related. So blood pressure is going to also have a role in how much urine you're forming. Your diet, your body temperature. If you're using diuretics, diuretics are going to promote you losing fluid in the urine. Diabetes, we kind of talked on that about when we talked about transport maximum. So if you have a high blood pressure, if you have a high blood sugar, then you're going to have high glucose concentration as it goes into the renal tubules. It's going to be so high that it exceeds your transport maximum, and then you're going to end up with glucose in the tubular fluid, and that acts as an osmotic agent, and it pulls fluid in. So then you end up urinating more fluid out. And then the amount of body fluid, or the amount of fluid that gets excreted by other means. So if there's a lot of sweating going on, or if you're vomiting, if you're losing fluid through some other mechanism, then that's going to reduce the amount of fluid that you're losing through the urine. So one thing that's important, you have to have a minimum of half a liter of urine per day in order to be able to eliminate the waste products from the body. So let's say that you just really were poorly hydrated and yet you're sweating a lot, so there's not a situation that's really promoting urine formation. You can't just not urinate at all. You have to produce and excrete at least half a liter of urine per day, because you do have waste products in the blood that you need to get out, and they need to get out through the urine. So if too little urine is produced and excreted, then those waste products will accumulate in the body. So these are some of the factors that influence urine volume. And these are all things that we've already talked about. There's nothing in here that's new information. So I'll just kind of go through them. I'm just going to go through one side because one shows the increase in urine volume, one shows the decrease. So I'm just going to go through the increase, and you can understand that the decrease is going to be the opposite. Okay, so if you get a decrease or a reduction in antidiuretic hormone, that's going to increase your urine volume. So let's look at that. What does antidiuretic hormone do? The more of it you have, the more aquaporins get put on that distal convoluted tubule, and the more water reabsorption that you have. So if you were to have an increase in ADH, you would have a reduction in urine volume. So this one's talking about the opposite. If you decrease your ADH production, you're not getting as much water reabsorption, which means more fluid or more water is staying in the tubular fluid, and then you're going to get an increased urine volume and increased excretion. Decrease in aldosterone. So aldosterone was the hormone that increased sodium channels in the tubule system. So we ended up with more sodium reabsorption, and then that consequently led to more water reabsorption. So if aldosterone goes down, that means we're leaving sodium in the tubular fluid. We're not reabsorbing it, which means we're leaving water in the tubular fluid, again, not reabsorbing it. So you're going to increase your amount of water that gets excreted with the urine. So lower aldosterone and lower ADH lead to higher urine volumes. Make sure that you can think these scenarios through. I really want you to understand what the hormones do, what the consequences are, so that you can kind of think through what's happening as opposed to just kind of memorizing it. Okay, an increase in atrial natriuretic peptide. So this was the one that kind of did the opposite. I think all the other hormones that we talked about, if you have an increase in those levels, it caused a decrease in urine production, but it's the opposite for atrial natriuretic peptide. So atrial natriuretic peptide was the one that led to an increase in glomerular filtration rate. And so as the fluid is flowing through the tubules really quickly, you have less opportunity for reabsorption. So basically more stuff stays in the tubular fluid. So higher A&P would lead to higher urine volume. Increase fluid intake. The more you put in, the more you put out. That's pretty simple. Increase blood pressure. So if your blood pressure is really high, one of the mechanisms that your body is going to use to try to get it back down to normal is to pull some of the fluid out so we can reduce our blood volume and reduce our blood pressure. When we pull that fluid out, we're going to put it in the urine and excrete it. And then diabetes mellitus. I talked about that one. I just mentioned it, I think, on the previous slide. We talked about how glucose acts as an osmotic agent. It pulls fluid in, so you end up excreting higher volumes of urine. And certain medications and alcohol act like diuretics, where they pull more fluid into the tubular fluid. And so then that leads to a higher urine production. So urine should always be some form of yellow. It can be anywhere from a light yellow to a dark yellow. The more volume of urine you have, the lighter it is going to be in color, whereas if it's a smaller volume and it's more concentrated, it's going to be a darker yellow. It should always be clear, assuming no microbes are present. And then they actually have a term for the normal smell of fresh urine, and that's called urinoid. So it has its kind of normal smell. Now, if you allow it to stand for a while, if you were to like pee in a cup and then leave it out for a while, eventually it will start to smell like ammonia, because you've got bacteria there that are going to start to convert the urea into ammonia, so it takes on that smell. Certain foods can alter the smell of urine. Undetected, uncontrolled diabetes mellitus can result in a sweet-smelling urine because you've got glucose present. And then urine usually has a pH of anywhere between 4.5 and 8. So just appreciate that that's a really broad pH range, especially when you compare it to something like blood that has to exist between 7.35 and 7.45. So this basically allows us a way to keep our blood pH within its narrow range, because any kind of acidity or alkalinity or anything we can push out into the urine, because the urine is cool to have this really broad pH range. Okay, so we've looked at every last little detail about creating the urine and the process of how we form it. So now we're basically just going to follow it through the urinary tract. So mostly what we've been talking about is everything that's been going on in here in the kidneys while we create the urine. And then from there, it's going to go into the renal pelvis and then enter into the ureters. It's going to travel down the ureters into the urinary bladder, where we're going to store it. And then when it's time for excretion, it will leave the bladder through the urethra and then get outside of the body. Okay, specifically looking at the ureters, they're long fibromuscular tubes. Again, their job is to conduct urine from the kidneys to the bladder. So you see them connected all the way up here from the hilum of the kidney, and they come in at the posterior aspect of the urinary bladder. When you get into the bladder, so the bladder is an expandable muscular container, and it's basically where we hold the urine until it's time for excretion. When the bladder is empty, it has this kind of inverted pyramidal shape. Looks like an inverted pyramid. But then as it fills with urine, it kind of stretches and it takes on more of an oval shape. At the inferior surface of the bladder, you see this area right here. It's kind of a triangular area. This is called the trigone, and this serves as kind of like a funnel. It allows the urine that's coming in to continually move towards the bottom of the bladder so that it can exit through the urethra eventually. The bladder's got 4 tunics, or 4 layers. The mucosal layer has got mucosal folds that allow for distension and recoil, so we can stretch as the bladder fills with urine, and we can recoil back to the original position as it empties. The submucosal layer lies just external to the mucosa. Then you've got the muscularis layer, which is made up of 3 layers of smooth muscle. Collectively, those 3 layers are called the detrusor muscle. So the detrusor muscle is the muscle of the bladder wall. At the neck of the bladder, there's an involuntary urethral sphincter. This is your internal urethral sphincter, so you can see that here. So this is the one, we've got two sphincters that regulate the passage of urine outside of the body. The internal urethral sphincter is involuntary. We do not have conscious control over that muscle. And then going back to the layers, you've got the adventitia, which is just that outermost layer. The urethra is a fibromuscular tube that is basically the connector between the bladder and the outside of the body. So you've got the urethral opening from the anterior-inferior surface of the bladder, and it goes all the way again outside. So you got these bundles of smooth muscle fibers that surround the mucosa, and they kind of contract, almost like a peristalsis, like we saw in the GI tract, and that helps to propel the urine out of the body. You've got two urethral sphincters. I kind of touched on this on the last slide, that restrict the release of urine until the pressure in the bladder is so high that we need to release those sphincters and allow the urine to be excreted. So in the last slide, we showed you the internal urethral sphincter. That's the one that's higher up. It's like kind of towards the neck of the bladder. That one's involuntary. We don't consciously control that one. It's composed of smooth muscle, and it surrounds the neck area of the bladder, and this one's regulated by the autonomic nervous system. Because autonomic nervous system, we do not voluntarily control. Do you happen to remember the name of the nervous system that we do control? I'll give you a second. Too late. It's somatic. Hopefully you got it. So the somatic nervous system we control voluntarily. So that means that the external urethral sphincter is going to be controlled by the somatic nervous system. I didn't even realize it was already written on the slide. So this one, the external urethral sphincter, is inferior, so it's further down from the internal urethral sphincter, and this one is controlled by skeletal muscle fibers. So we always control voluntarily our skeletal muscle fibers. So this is the one that we consciously control when we decide it's time to urinate, and this allows us the ability to control when we do so. And it's also the muscle that we are teaching children to use whenever we potty train them. The male and the female urethras are different in their lengths, and that has to do with the anatomy of the urogenital structures. So the female urethra has one single function, and that is to transport the urine out from the body, from the urinary bladder to the body. It's very short relative to the male urethra. It's only about 4 centimeters long, and then it opens to the external urethral orifice. So here's your bladder. Around the neck of the bladder, you have your internal urethral sphincter. There's a very short urethra, and then your external urethral orifice, where it actually leaves the body. So the male urethra serves a couple of different functions. It's actually part of both the urinary system and the reproductive system. And that's because it serves as a passageway for both urine and semen, though not at the same time. So it's about 19 centimeters long, and it's broken down into 3 segments. You've got the prostatic urethra, which you can see here is going right kind of down the center of the actual prostate gland, which is this thing surrounding it. So the prostatic urethra kind of starts right below the bladder and then just runs throughout the prostate glands. You've got prostatic ducts that will contribute fluid to the prostatic urethra. And then the next segment is the membranous urethra. This is the shortest and the least dilatable portion of the male urethra. It's going from just below the prostatic urethra through what's called the urogenital diaphragm, which is a structure right here. And then the longest part is called the spongy urethra, and then it runs through the penis, and it's surrounded by the erectile tissue of the penis called the corpus spongiosum, and it will go all the way until you get to the external urethral orifice, where the urine can then exit the body. Urinary tract infection, so this is going to be when bacteria which is either most commonly going to be E. coli, or sometimes you can get fungus or yeast there. And so it gets into the urinary tract, and there it's going to start to multiply. Women tend to be more prone for urinary tract infections because the opening to the urethra is in closer proximity to the anus than what you find in males. So some of that bacteria from fecal material can get moved into the urinary tract, and then it can start to reproduce. So sexual intercourse or the use of a catheter, those can increase the risk of urinary tract infections, although, like I said, a lot of times they come kind of spontaneously from contamination from either fecal material or some other bacteria has made its way in. Okay, so UTI often develops first in the urethra. So if you have inflammation there, it's called urethritis. And then if you kind of just keep moving up the tract, if it spreads into the bladder, it's called cystitis. Occasionally the bladder from an untreated urinary tract infections can make its way all the way up to the kidney, in which is called pyelonephritis. Some of the symptoms are going to include difficult or painful urination, that's called dysuria, frequent feelings of needing to go or frequent urination, kind of this uncomfortable pressure that you have kind of in the region of the bladder. If the infection spreads to the kidney, you're going to end up with kind of back pain, where the kidneys are going to sit kind of posteriorly there. You can diagnose the urinary tract infection through a urinalysis, so you can look and see if you have leukocytes or blood or bacteria or fungi or any of that, because that normally should not be present in the urine. So if it's there, it's a good diagnostic tool to look for urinary tract infections. And so then we can just use the appropriate therapy. So if you've got a bacterial infection going on, we've got antibiotics, and then we've got antifungals if you were to need something like that. So urination is the expulsion of the urine from the bladder. It's called micturition, it's called urination, and it's called voiding, because why would we only have one name? So there's two reflexes associated with it. You've got the storage reflex, which is where we're going to actually hold it, and then the micturition reflex, when we're going to let it go. Those reflexes are going to be regulated by the sympathetic and parasympathetic divisions of the autonomic nervous system. So the sympathetic division is what's going to be responsible for that storage reflex or holding it in, and then parasympathetic is going to be responsible for the micturition. So just think of like rest and digest. Whenever you're digesting, you're just kind of all of those normal processes that need to happen, they like to happen when we're in parasympathetic mode. During sympathetic mode, we're in fight or flight, we don't want to worry about all those maintenance activities. We want to focus on heart, lungs, blood pressure, skeletal muscles, and all of that stuff. So we're going to actually be working on holding it under sympathetic nervous system. Okay, so when the sympathetic nervous system is stimulated, it's going to cause a contraction of the internal urethral sphincter, and it is going to inhibit the contractions or otherwise relax the detrusor muscle. So if you think about what would cause you to urinate, it's going to be the relaxation of that internal urethral sphincter and the contraction of the detrusor muscle, because that would push the urine out and the sphincter's open, so it's going to allow it to pass. So that's only going to happen under parasympathetic activation. Sympathetic activation is going to do the exact opposite. We're going to relax the detrusor muscle and contract that internal urethral sphincter in order to hold it in. Okay, so again, the parasympathetic then is going to actually contract the detrusor and relax that internal urethral sphincter so that the urine can come out. And then eventually gets to the external urethral sphincter. That's the one that was composed entirely of skeletal muscle. And so we control that voluntarily. So that's what actually allows us then to go. So a little more on the specifics of the reflexes of urination. So we've got the storage reflex, which again is under sympathetic domination, and then the micturition reflex, which is under parasympathetic domination. Okay, so storage reflex, we're basically storing it. So what's happening is we're producing urine in the kidneys. They're going to move through the ureters, and they're going to start to fill the urinary bladder. And so based on the sympathetic inputs, you're going to get a relaxation of that detrusor muscle so that the urinary bladder will relax and allow stretching so that we can accommodate the influx of urine that's coming in. And then it's going to cause the contraction of the internal urethral sphincter so that the urine stays in the bladder as opposed to coming out. So the micturition reflex is the one that's going to happen whenever parasympathetic activation prevails. So this is going to be the process of urination. Again, it's caused predominantly by the autonomic nervous system, but there is that component of the somatic nervous system that's related to that external urethral sphincter, and that's the one that we're working on in potty-trained individuals. So when the volume of the urine retained in the bladder reaches about 200 to 300 milliliters, the bladder starts to become distended, so it starts to stretch. And you have baroreceptors in the bladder that become activated. So baroreceptors, they become activated as stretching in organs happens. The baroreceptors send nervous signals through the visceral sensory neurons up to stimulate the micturition reflex that happens in the pons in the brain. And then the micturition center sends nerve signals down through the spinal cord, through the parasympathetic nerve fibers, and those are going to cause the detrusor muscle to start to contract, and it's going to cause a relaxation of the internal urethral sphincter. So the bladder is contracting, the sphincter is open, micturition can occur. So in infants or individuals who are not yet potty trained, that's the point where urination would happen because they don't yet have control of that secondary external urethral sphincter. If you do, if you're talking about like an adult individual or an older child that's already potty trained, then there's that kind of added component where then you have to have the somatic nervous system relax that external urethral sphincter in order for urination to happen. Two kind of common issues that you have with urination are incontinence and retention. So incontinence is the inability to voluntarily control urination. It's just kind of come out without our ability to fully control it. So this can happen as a result of childbirth if you have weakness in those pelvic floor muscles. Stronger than normal detrusor muscle contractions. So normally the detrusor muscle of the bladder has these kind of mild contractions just to kind of help it get out. But if you can imagine that detrusor muscle really starts doing these really strong contractions, it's really just going to kind of force the urine out that way. That's called urge incontinence when that happens. And then also it can be a secondary result of certain medications. So uncontrolled urination can happen also when an individual is frightened. So this is like a whole different kind of reflex on its own that I'm not really going to go into. And then the other issue is retention, which is the inability to eliminate the urine normally. So now you want to get it out and you can't. So causes for retention that can happen if you've been under general anesthesia. So it kind of slows all of the processes down and it makes it hard to fully eliminate. An enlarged prostate gland in males. So if you go back to that diagram of the urethra, and the prostatic urethra went right through that prostate gland. So if that prostate gland is enlarged, it's basically going to impede upon the space of the urethra, and it kind of pinches it off to an extent. So that makes it harder then to get the urine out. If necessary, you can insert a catheter that can be placed through the urethra to the bladder, and that will help to get the urine out.